Hello everyone, hope you're all doing well. Myself Seema Tambe, and today we are going to talk about what is indicator. Okay, let's see what is indicator, guys. Indicator. So it goes by name. Okay, indicator, it indicates something. Okay, it shows something. Okay, so what is indicator, guys? The substance, okay, which the substance, or substance is nothing but what? It's a chemical substance. The substance which changes its color in acidic and basic medium. Okay, what is indicator, guys? The substance which changes its color in acidic and basic medium, or acidic and basic solution, you can see. What? The substance which changes its color in acidic and basic medium. Okay, indicating that particular solution is acidic or particular solution is basic. How you get to know? By putting the indicator into it. Okay, like for example, we'll see. If at all this is acid, okay, this is what, guys? This is acid, okay, and this is base, okay, this is what, guys? This is acid and this is base. Okay, we are going to talk about the types of an indicator, but let's just see how, just to know whether the solution is acidic and basic, and showing the example. Okay, let's say this is acid, this is base. Okay, so if you take a blue litmus, what if you take a blue litmus and you dip into this acidic solution? What I said, if you take a blue litmus and you dip it into the acidic solution, or you don't know whether the solution is acidic, you just take the blue litmus and dip it into it. If the blue litmus turns red, okay, if blue litmus turns red, then you can, it's a confirmation that the particular solution is what? Acidic. Okay, and if you take a red litmus, okay, and you dip it in this solution, okay, if this red litmus turns blue, okay, if you're taking red litmus and you're dipping it to this basic solution, if it turns blue, if red litmus turns blue, then it shows that the particular solution is what? Base. Okay, so see, that's how you get to know whether the particular solution is acidic or basic. See, we don't, for example, we don't know that the solution is acid or the solution is base. Okay, you take the solution, okay, you take the solution and you take this litmus paper and dip it into it. If the blue litmus turns red, it shows that the particular solution is what? Acid. If you take a red litmus and you dip it into the solution and if it turns blue, then it is a confirmation that your particular solution is what? Base. Okay, so hence it is called as an indicator. It indicates whether your solution is what? Acidic or basic. Okay, now this we are going to talk in detail. Okay, so did you got to know what do you mean by indicator? Indicator is what, guys? It's a substance which changes its color in acidic and basic medium. Okay, indicate. Okay, it indicates. Then there are types of indicator which we are going to see. Okay, the types of indicator, indicators types. Okay, so there are three types of indicator, guys. Okay, there are three types of indicators. Okay, first one is what? Natural indicator. Second one is synthetic indicator. Okay, I'm not going to write indicator in, okay, indicator, I means indicator. Okay, natural indicator, synthetic indicator and olfactory indicator. Okay, indicator. Fine. There are basically what, guys? Three types of indicator. What are those? Natural indicator, synthetic indicator and olfactory indicator. Okay, what do you mean by natural indicator, guys? The indicator which you derive from natural source. okay, which is what, plants or animals. Okay, that particular thing is called as what, natural indicator. The one which you get or which is derived from what, plants or animals. Generally plants, okay, which is derived from plants, you can say. Okay, sorry, not animal, it's plant. Okay, the indicator which you get from plants, okay, or from nature is called as a natural indicator. Synthetic indicator which you derived or which you prepare in laboratories. Okay, olfactory indicator, we'll talk about it. Okay, what do you mean by natural indicator? It is like which derived from plants, and synthetic indicator which you get it from laboratory or which you prepare in laboratory. It's man-made. Okay, so what a natural indicator? There are basically, you know, 3 types, which is what, first one which we have seen just now, litmus, the litmus paper. It's a first indicator. Second one indicator is what, you know, your turmeric. Okay, which we use in our daily purpose. And China rose. Litmus paper is what, guys? Litmus paper, or you know, litmus is basically, you know, it is extracted from a plant. It is extracted from plant called lichens. Okay, it is extracted from a plant named lichens. Okay, or lichens or lichens, whatever you say. Okay, it is basically derived from plant. Okay, so what they do is they just extract litmus from plant. Okay, and if you just dip it into water, that particular solution becomes, you know, purple kind of, purple color. Okay, and nowadays like 2 form, which is what, red litmus and blue litmus. So this red litmus and blue litmus which you use in your laboratory. Okay, so that is what, that is a plant, that is from plant source. Okay, which is what, red litmus and blue litmus. This is a indicator. Red litmus and blue litmus paper. It's an indicator which is natural indicator. Turmeric indicator, the turmeric which you get in your house. Okay, what you do is just take that, okay, turmeric, and just put it onto some paper. Okay, and okay, and then you drop for example soap onto it. Okay, what you will see, that soap, that particular solution will become, or that the spot on which you have added the soap, that will turn into red color. Okay, has soap is what, base, and you are adding it, this turmeric, what, it's an indicator, so has it is base. So if you are adding that drop onto that turmeric, you know, that turmeric paper, you can see that spot will turn red in color. Okay, indicating that solution is base. Okay, soap is base, so soap, the whenever you add indicator. Okay, or you can say when you take soap solution and you drop it onto this turmeric paper, the soap or the indicator turns into red color. Okay, indicating that your solution is what, base. Okay, so this is the second indicator, which is turmeric. Third is China rose. Turmeric. Third is China rose or shoe flower, you see. Okay, what you need to do, you need to take that shoe flower and, you know, boil it, and you just put it into the boiling water. Okay, and the whatever solution you're going to get, that is basically used as an indicator. Okay, fine. So these are 3 types of indicator. What? Litmus paper, turmeric, and China rose. Fine. Now the synthetic indicator is what, guys, which you obviously know. Hydrochloric acid, H2SO4, right? These are all the synthetic indicator. Sorry, I'm so sorry. The synthetic indicator is what? Phenolphthalein. Phenolphthalein indicator, phenolphthalein, you can say, phenolphthalein, yeah, it's a phenolphthalein indicator, and methyl rose. These 2 are synthetic indicator. What are synthetic indicator? Which is derived from laboratory, first thing, and example is what? Phenolphthalein and methyl orange. Okay, now what happens, you know, guys, the Phenolphthalein, it's what? It's a chemical substance, okay, which is what? An indicator, synthetic indicator. Methyl orange is also an indicator. So if you take acid, okay, and you add phenolphthalein into it, okay, the phenolphthalein basically remains colorless in acidic medium, okay, indicating your solution is acidic, okay. If you are taking base solution, okay, and you are adding a drop of phenolphthalein into it, that solution will turn pink, okay, indicating that your solution is what? Base, okay, fine. So this is what phenolphthalein, which is when you're adding it to acidic solution, it basically remain colorless, and if you're adding in basic solution, then it turns pink, indicating that solution is basic. Now with methyl orange, if you take acid and you add methyl orange, that if the solution is acid, that will turn into yellow color. If the solution is base, that will turn into red color. Okay, if the solution is what? Acid, then it is going to turn yellow color, indicating that's an acid, and if it is a base, then it is going to turn into red color. Okay, so these are the two indicators which are used in laboratory. There are many indicators, but this is two, like 11th and 12th, or you can say, sorry, in 10th, 9th, 10th probably now. So it's like these are two indicators are generally used, like phenolphthalein and methyl orange. Okay, an olfactory indicator. What do you mean by olfactory indicator, guys? The indicator basically which changes its smell and taste. Okay, when you're adding into acidic or basic solution. What is olfactory indicator? The indicator which changes its taste or smell. Okay, what is olfactory indicator? It's an indicator which changes its taste. The indicator which changes its taste and smell when added into acidic or basic solution. For what are the examples? Is like onion, cloves. These are the indicators, which is called as olfactory indicator. Means what? When you're adding it into acid, the smell changes, and if you're adding it to the base, the smell